So welcome everyone. Thank you for joining us this afternoon for our next in our Let's Talk About It series. I'm Rabbi Dan Moskovitz, and I want to thank Dr. Tirosh Shapira for joining us this afternoon for this conversation about COVID-19 and the work that he is doing on the virus and on drug treatments and research with the virus. Tirosh, welcome. We're glad to have you.
Thank you. It's a pleasure to be here.
So before we jump into the questions that I want to ask you, can you tell us a little bit about you and how you found yourself at UBC doing this kind of work? What brings you to British Columbia and this work? Are you there?
But I think pretty much I can't talk about myself. I guess that was where the question was going before the
comment. I lost you for a second. But just so we know who you are a little bit.
Yeah, so that's a big question, but I'll try to keep it short. I am a PhD in biology. I've done my studies at the University of Queensland, where I researched how to protect grain storages from pest insects. And from there, my wife followed me from Israel to Australia for my studies. So I followed her over here to Canada, keeping it equal. She found a research lab, a great research lab here at SFU. So I was looking for somebody to continue my education with here in Vancouver, and found a great opportunity researching tuberculosis at UBC. I've been doing that for the past 2.5 years, and very happy with the outcomes of this research. But as we all know, COVID struck, and I felt that my expertise and skills can be used for developing drugs against SARS-CoV-2. That's when I decided to move and shift into this research. And and I've been enjoying it since. About myself more generally, I was born in Israel, but grew up in the United States. My parents are academics, so we were moving around a lot. Afterwards, I did most of my education in Israel before my PhD, which was in Australia.
Wow. And it was your idea to get involved in the COVID research, or did they draft you for this?
No, it was my idea. The primary researcher who got funded for the original research was looking for people. I did hear through the grapevines that they were looking, but it was my decision to present myself and say, listen, I have this particular set of skills that I know you're looking for. And of course, we had a very good collaboration since and a good relationship. So it was all very positive.
And I am going to get to the questions that we had talked about, but I'm just curious to know, so how many people are working on COVID, vaccine and drug treatment research in Canada right now, or in UBC?
Yeah, so surprisingly, not a lot. It's a complex answer, but mostly the access to the virus is very restricted. Only the Public Health Agency of Canada can give authorization to research facilities to deal with the actual virus, because it is so contagious, because it is so deadly to the people that it does infect. Then regulation and work on this virus are quite strict, and they can only be done in facilities that are determined to be contamination level free facilities. Mm-hmm. Now, luckily for us, this is exactly where tuberculosis research is done as well, for similar reasons. And so UBC has such a facility, and we've been working in the past couple of months to accredit it for SARS-CoV-2 research with PHAC, and have gotten licensing and gotten the virus here to UBC to do the actual drug discovery research. That doesn't mean that we are the only ones working on it. There are other groups that are working with the live virus at the University of Toronto, where they isolated the virus, and in NML, National Microbiology Laboratory in Winnipeg. And there are a lot more researchers without working on the virus itself. That can be both in the field of biology, using models that serve as proxies. They don't serve as such great drug development models because if you develop a drug against a similar virus, a lot of times you don't end up with a drug that can actually treat the virus. But there is some invaluable information that can be gained from these models. So I think if there are 10 research groups in Canada, if I need to eyeball it, who are working on SARS-CoV-2 itself, and we are the only ones in BC who have that license, then there's going to be about 200 biologists working on it and about 2,000 people working on the economics, working on social studies and how stress responses in response to COVID-19 have changed. A lot of research that is not directly related to COVID-19, but COVID-19 is playing a big part of research of people who have been studying the social sciences as well as the biologists.
Mm-hmm. So getting to some of the questions that we had talked about beforehand, is working on COVID-19, is doing drug development on COVID-19 different from work that you've done or that others do on other pathogens, and how is it different?
Okay, so again, there was a bit of a cutoff with the question.
The question is drug development for COVID-19 different than for other pathogens, and how is it different? How is developing drugs different for this as opposed to other things?
Yeah, no, that's an excellent question. The main issues with this pathogen, one issue that who can't, which obviously creates some barriers in the speed of developing drugs. If we have had more people, then more results would have happened faster, obviously. But also, this virus is unique because many viruses, but this one in particular, have a different type of genetic material than we have. Because viruses aren't living organisms, they're expert saboteurs. They know how to use us in order to make more of them, but they're not living organisms per se. So a lot of the restrictions that living organisms have, like having DNA as your genetic material, like specific components inside each and one of your cells, that virus doesn't have that. And the variance of genetic material it has is much less stable than the one that we have, which makes it very easy for it to mutate. So if we discover one drug against COVID-19 and we give it for a long time, it's very likely that we'll see resistance that is developed against this one drug. And so when we are developing a drug, we should also be thinking about what is the half-life, or how long will it take before this drug that we're developing can no longer be used. The way that it pertains to my research is that instead of looking for one drug to treat COVID-19, I'm looking for 4. We're trying to create a cocktail, similarly to what is applied when treating HIV, for similar reasons. HIV is also a rapidly mutating virus. And so if there's a chance of 1 in 10 that the virus will mutate, and there's more than 10 viruses in the person, when we give the drug, you can be pretty sure that there will be a resistant virus in there. However, if we're adding 3 or 4 drugs that each one of them has a 1 in 10 probability, then suddenly the chance that the population of viruses will have a mutant that is resistant to all of these is going to be very unlikely. Approach. We're not just looking for a drug, we're looking for drug combinations, and drugs can interfere with each other. You can have 2 excellent drugs that each one works individually, but together in a combination, they just negate each other's effects. So that's part of the complexity of looking for cocktails rather than looking for specific drugs. And because this is an RNA virus, or the specific type of virus that mutates more rapidly.
Tirosh, we lost your audio there for just a little bit. You said because of the virus that mutates
Okay.
more rapidly, and then we lost you after that.
Yeah, because the virus mutates rapidly, that is why it complicates matters, and we can't only rely on one drug to be given to patients. We have to create combinations. Otherwise, very, very soon we will see drug resistance emerge.
Got it. So, I mean, I know that from everything I've read, that the development of a vaccine or drug treatments generally takes, years and years, if not decades. And, we're trying to talk in months and, in 18-month, outside kind of numbers, which, everybody has told us is incredibly accelerated. But, we're in the midst of living with this. So how long can we expect it to take for a new drug to reach patients, or in this case, new drugs, as you talk about the cocktail?
Yeah, so it'll be hard to put an exact number on it. There's a lot of politics involved with FDA review, or when can a drug reach patients. With previous drugs that have been developed for rapidly emerging diseases, like the Ebola outbreak of several years ago, we saw about 18 months before a drug, or a vaccine in this case, from being developed. Doesn't mean a drug is commercially available. It means it's in clinical phase trials, and one of the clinical phase trials that is done is to give the drug to people who need it. So theoretically, if tomorrow I'm extremely lucky and my test results come great, and I have 4 drugs that can completely cure SARS-CoV-2, in about 3 or 4 months, this can be given in a clinical phase 1 trial to participants who are in need of it.
You've frozen for us again here.
Yeah, but excuse me.
So you froze. Given to participants who are in need of it, and then we lost you there.
Yeah, so for participants who need it, it can be given in 4 to 3 months, but then instead of 5 years, which would be the normal expectancy, or even 10 years, if it's not accelerated, because it is accelerated and there's such a need, we might be looking at less than that. Within a year or so, it will be an available commercial product that people can use. But there are other issues that complicate it that I am intending to talk about a bit later, which are production lines. So producing for drugs and distributing them on a massive scale and the speed in which they're needed. Even if I find a drug combination tomorrow, even if the FDA reviews it and approves it, we're talking about 6 months just to get the process going. There was a recent story with famously President Donald J. Trump touted this treatment against SARS-CoV-2. The US stockpiled on massive amounts of it, and when the FDA withdrew its recommendations, then there was not much to do with the extra stockpile, and it's now taking space in the United States emergency reservoirs, storages. At the same time, this drug stockpiling reduced the amount of hydroxychloroquine for people who actually needed it. But for speaking about my drugs, when they come and are fully developed, even something like the glass vials that were used to store these hydroxychloroquines are not in massive enough supply for us to keep on stockpiling on drugs in case we will find them effective.
Your video's cutting out on us again there. Your audio. You there still?
Okay.
So I know you have a presentation that you've prepared, and I've been pummeling you with questions, and I'm sure we're going to have more questions after your presentation. Do you want to go to your slideshow now, and we'll pick it up from there? Okay. So you're able to share it on your screen, I believe, with the share button?
Yeah, no, I would like that. And again, this will, some of the questions that we discussed now are going to be described a bit more in depth in the presentation. So yeah, I think it will help visualize what I've been talking about, especially if I froze a bit and it wasn't easy to understand exactly what I was saying. Yeah, so I can start the presentation now.
Great.
So all the introductions were made already, but I want to frame the questions of what this talk will be discussing and what it will not be discussing. So I am going to talk about SARS-CoV-2 in the background and of what infectious diseases are, how do we regularly discover drugs, and talk to you about what is currently done at the University of British Columbia. I do want to emphasize, though, that anything that I say here is not clinical advice. Obviously, physicians and medical doctors who specialize in internal medicine and infectious diseases are those who should be giving these types of advice. But I'll bring a bit of my understanding of the biology and how it reflects in the news cycle that we're seeing every day now. So infectious diseases have been plaguing mankind for as long as we remember. As you can see on the map on the left, currently it's plaguing poorer countries and developing countries more so than it does developed countries. But still, even if we take developed countries into account, infectious diseases are responsible for about 1/4 of world mortality based on the numbers from 2004 by the World Health Organization, but that number hasn't changed significantly. What has changed is if you look at specific countries, obviously in Canada, we know not 1 of 4 people who have died from infectious diseases, and sadly more than 1 in every 4 people that has died would die of cardiovascular diseases. But that just emphasizes that in places like Sub-Saharan Africa, like Southeast Asia and mainland Russia, infectious diseases and parasitic diseases are taking a much larger burden. And in those countries, approximately 1 in 3 people will end their life because of a preventable infectious disease. As I said, as part of our history, they also shaped who we are as people and how our culture developed. We see that infectious diseases and their burden are tightly linked with how many kids a family will decide to have. In countries that have large mortality due to infectious diseases, we see a lot more children being born. We also see more xenophobia in those communities because you know what kind of infectious diseases are in your community, but somebody coming from outside is very likely to carry an infectious disease with them. Infectious diseases also affected the art, and this is one of my favorite painters, Munch, and his famous painting, The Scream, depicts part of the existential crisis that he felt after both his mother and his sister, depicted in the picture above, have died of tuberculosis. It also affected La Traviata, the famous opera, is written about a young socialite who is dying of tuberculosis and wants to live the last days of her life in the most lucrative way that she can. And infectious diseases also affected politics and religion, famously with kings and Christendom claiming that through divine right they can cure illnesses. The infrastructure of kings and the feudal system were very much strengthened by people coming up looking for what was called the king's touch, which was claimed to have healed leopards and heal other types of infectious diseases. There are many agents that can cause infectious diseases, and each one of them will create a very different type of disease. So we know bacteria like tuberculosis that I just discussed can cause infectious diseases, but there's also other types of diseases like malaria, like thrush, like the mad cow disease. And some of these agents aren't even living organisms. So bacteria and fungi, we know to be alive. We even rely on them for a lot of our food production. But viruses and proteins, and especially viruses, which we're focusing on now, aren't even living agents, but still they can spread between us and cause terrible diseases. So what are viruses? Viruses are a large array of different agents, each one of them with unique characteristics, and each one of them relying on their host in order to replicate themselves and create more copies. Because, as we discussed, viruses are not living organisms, they vary greatly with what kind of genetic material they use. Do they use proteins or do they use fatty acids to protect themselves from the environment? And we have similar families of viruses like SARS and the common cold, which basically are very similar. So if we look at what is similar between SARS and the common cold, we would see about 90% identity in the sequence that is coding to what this virus will do and how it will be made. Even more so, we would see similarities between SARS-CoV-2 and SARS-CoV-1, but the infection and the disease that it is causing are quite different. However, from knowing the common cold well, from knowing SARS-CoV-1, from knowing Middle East respiratory syndrome virus, which are all part of what we call coronavirus family, we can predict a lot of the ways in which the biology of SARS-CoV-2 will play, and that is exactly how we discovered remdesivir. As we have previously known, it is active against MERS, or Middle East Respiratory Syndrome virus. So infectious diseases are terrible and they've been plaguing mankind, but we have also found ways to combat them. So if we keep on talking about TB, because that's what I've been studying for 2.5 years, so I know a bit more of the history and the background, we can see here a graph of how TB instances declined in Europe over the 20th century. And what you can see is that a lot of advances have happened. We identified, or Robert Koch identified, the TB bacillus in 1890. We discovered ways to detect TB. We even discovered antibiotics against TB. And each one of those slightly decreased the downwards trend of infections of tuberculosis. We also had some devastating events like the world wars that created very poor sanitary conditions that didn't allow for proper medical treatment, that also changed the downward projection of tuberculosis. But what I want to highlight in this is that discovering the virus or discovering a cure against the virus is not what is needed in order to get rid of the virus. It can definitely help, and it will be of great help for the people who need it. But overall, there are other factors who control the epidemic and its effect much more than any drug or specific treatment. So when we're looking at infectious diseases, first of all, it's politics and economics that have the most control over the spread of the disease. And we do see that the increase in the modernization and the industrial revolution that swept through Europe has greatly improved the diets of the people, their ability to get good medical treatment. Even after a certain point in the Industrial Revolution, their ability to get fresh air. All of these were great factors in increasing our ability to get rid of tuberculosis. Sanitation and containment, or the fields of epidemiology, are another great way to get rid of viruses, and they're very effective. So by contact tracing or figuring out who has talked with who and might have delivered an infectious agent, by making sure that, for example, our waters are clean and treated well, that our outhouses are outside, and that there's toilets in every household in every nation, we can greatly reduce the burden of infectious diseases against SARS-CoV-2 specifically, and all infectious diseases in general. not less important, but less impact on the spread and the outcome of a pandemic are the treatment, which is what I am developing, and microbiologists and pharmacologists are working hard on developing, then also the individual treatment like pedagogy and specific treatment in medicine of somebody who is currently suffering from severe infection with SARS-CoV-2. While they are very important, each individual case has little to do with the overall global spread of SARS-CoV-2. That being said, some education campaigns have been extremely effective in protecting against infectious diseases, most famously against HIV. So promoting safe sex and promoting the use of condoms has greatly decreased HIV, and that is purely an educational campaign. Some treatments, some drugs have completely eradicated infectious diseases, including viruses, like measles has almost been eradicated, and we're now in the process of eradicating Ebola, though there has been some outbreaks recently in sub-Saharan Africa that are a bit concerning. But treatment can also greatly affect the spread of an infectious disease. However, at the global scale, it is more sanitation and containment that we should be looking for immediate response to this immediate threat. So this was a bit of a lot, so I want to summarize and maybe give a chance for questions about this. But understanding SARS-CoV-2 as researchers comes from our understanding of similar viruses. We know from similar viruses that the best way to defeat them is through social adjustments, which is what we're all doing very well. And despite SARS-CoV-2 being a present threat, it will pass, and regrettably other infectious diseases will also emerge after it, just like Ebola emerged several years ago and SARS-CoV-1 emerged before that. We are in an era where plane travel is more easy than ever, not specifically in these days, but an infectious disease, despite originating maybe in Ontario, can find its way very quickly to Vancouver and can find itself very quickly to China. Another infectious disease that emerged in Russia can similarly travel very quickly. So these diseases are not restricted to a specific area, and that is what is promising that a new infectious disease will come. And the more we know about these families of bacteria and viruses that are causing these diseases, the more prepared we are when they come with medical solutions that can stop them from becoming epidemics. Are there any questions that people want to use the opportunity to ask? Or if not, we can move forward and discuss drug development and modern drug development.
Well, Tirosh, let me just pause here for a second to offer people the opportunity to put your questions in the Q&A button. Those that are joining us on the webinar down at the bottom, and we'll process those and bring them to Tirosh as we go. But why don't we keep going with your next slide or two, and that we'll collect some more questions and ask you them, okay?
Yeah, perfect. Yeah, I would love for this to be more of a discussion. Like, I'm skilled at giving presentations, especially to undergraduate students, not so much to congregations, but this is an opportunity to really communicate and ask questions while we're talking. So any questions that you have will be great.
Okay. Yeah, so as we're waiting for those questions, you know, let's keep going just a little bit, and then we'll ask some more, okay?
Okay, so I'll drop into the next stage, which is how do we develop drugs nowadays? So drug development has been going on for about 100 years. And the classical approach is what my father has been doing and what other biologists have been doing. They've been looking underneath a microscope, trying specific treatments on bacteria and viruses and fungi, and trying to see what drug can affect them. However, the modern approach instead of being top-bottom or discovery-based and then trying to develop a very good pharmaceutical from what we find in our lab, the modern approach is very, very different. The modern approach thinks about all possible compounds and is less concerned about the biology. Instead of figuring out the biology and saying, oh, we know this virus behaves this way, so this compound might be very effective against it. What the modern approach does, based on our huge advancements in mechanization, automation, computing power, it just basically says our goal is to get a drug and we don't really care what the biology is. So let's throw all that we have on the virus and see what sticks. It's a long process and it requires a lot of design and good execution, but in the end, the modern approach is proving much more effective than the classical approach of drug development because it allows us a lot of things that the classical approach usually does not allow. It minimizes the research bias because usually biologists will love their specific molecules that they've been researching for ages, and they will tend to favor them and not look outside the flashlight where they were looking to see what else is out there. Also, a lot of biologicals that we find in the lab are working excellently in a petri dish, but once we test them and try to make drugs out of them, we discover they have very poor pharmaceutical properties. So instead of starting with good biologicals and trying to make good drugs out of them, the classical, the modern approach compared to the classical takes already good drugs, and we'll discuss which ones these are, and tries to see which one of them is going to be effective as a biological. And because we're testing a lot of drugs, we have a lot more statistical power over experiments that are done in the lab, which gives us a lot more confidence in our predictions and in the drugs that we forward to clinical trials. And indeed, the modern approach of throwing everything at it has been proven to be more effective in developing compounds, and that is why drug companies are doing it left and right, and so are universities nowadays, because drug development is a very expensive and very lengthy process, and so it's basically trying to figure out what works better, and this modern approach seems to work better. It doesn't mean that the biology is not important. So when you test a lot of drugs against an agent, a biological agent, an infectious agent, the question that you're asking is of paramount importance. How good your model is. And we kind of referred to that earlier in the conversation with Rabbi Dan. If you do not investigate the SARS-CoV-2 virus, but instead try to develop a drug to SARS-CoV-2 by developing a drug against the human common cold virus, which is similar, you might very well get a very good drug. However, more likely than not, you won't. So having a good test model and a most accurate model that you can get in a lab, that will determine how good your outcomes will be. Or there's a famous saying in computer sciences, garbage in, garbage out. I like to reverse it and say gold in, gold out. If you have good, sound biology, you'll get good, sound compounds that are good pharmaceuticals. So what kind of compounds do we throw at the virus? So first of all, obviously we throw antivirals that are already established, because if they work against one virus, there's a chance that they'll work against the new virus. We also test FDA-approved drugs. These are drugs that have passed development, they have passed review, so we know they are safe, or as safe as drugs can be, and that greatly shortens the approval time by an FDA review. Also, because we know a lot about them and we gathered a lot of information about them, we know exactly their half-life in the body, what doses should we give of them, what doses shouldn't we give of them, how can we give them effectively? Is it via a pill or maybe injections? So FDA-approved drugs make great candidates for such compounds to start with our screens with. Also, drugs that are in clinical trials for the same reason have already been studied for their pharmacology properties. And so they're better candidates or better drugs than any random compound that we'll throw at it. Last but not least are natural products derived from animals, from other microbes, from trees, from mammals. Traditionally, all the antibiotics or most of the antibiotics that we developed have been previously discovered by isolating them from other organisms. So just like we have been living with viruses and infectious agents, so did other animals, and so did other plants, and so did other microbes. So they have already done a lot of the work for us. They have developed antibiotics and they have developed antivirals. And so taking a compound that we isolated from natural products is a great source of potential compounds. And because they were made by living organisms, they also usually have very low toxicity. So again, they make for better drugs than anything we just isolate from nature, from a rock or from clay, for example. So I talked a lot about the theory, but how does it actually look like? So on the left, you can see multiple plates, and in each one has about 100 wells. And in each wells, we put a different drug and test it against the virus that we're looking at. And with advanced screening methodologies, we really capture and track is the reduction or increase in the amount of viruses in the treatment that we're looking at. And you can see how it looks on the right. In this case, this is my results from Mycobacterium tuberculosis. That is the middle panel that says MTB on it from the top. And you can see that in the top image, in the top central image, you can see a lot of staining or a lot of bacteria when we don't treat with any drug. If you look one panel underneath, you can see an antibiotic that is known to work against tuberculosis almost completely decreases the prevalence of the bacteria. And then what we simply do is tell a computer which one of the wells from the screen on the left looks more like the antibiotic than it does the non-treatment. And because we have very strong computing powers, that is a very rapid analysis and a very powerful one from statistical perspective. So we talked about drug development and gave an example of how it happens, and the exact same process that I developed against tuberculosis is now used by me against and others against SARS-CoV-2. And all of those are encapsulated under what we call preclinical trials. Usually they start in a lab and then they progress into animal testing, usually in mice. Mice are a very poor model for SARS-CoV-2 because they don't get naturally infected with SARS-CoV-2. So we face a bit of a problem before we get to clinical trials because we don't really have good animal models to test them with, but our lab has a nifty solution to that, which I'll be happy to talk about in a second when we're discussing my research in more depth. Then we have clinical trials that are traditionally separated into 3 stages. Each stage takes about a year, and I can see from the standard approval process, the top one, as we discussed with Rabbi Dan, this is a very long process usually, but there are cases of expedited drug development, and the shortest ones that we know, drugs have been available, fully available, in about 2.5 years from starting to look for them. After the clinical trials comes a stage of FDA review of the outcome of the clinical trials, how rigorous the data was, how severe were the side effects that were discovered, was the methodology that was done well enough. And without FDA approval, nobody really is going to treat anybody with a compound just based on preclinical or even clinical trial information. The last process, as I also alluded to, is production and distribution once the FDA does approve a drug, which is also quite a lengthy process, about 6 to 18 months based on the complexity of the drug, of the compound, and the complexity of distributing it to everybody who needs it. So again, I'll do a short summary, and maybe this would be a good opportunity if there were questions from the previous section, and maybe if there's questions from this section to also join in. Then drug development has greatly advanced, and we are more capable now than we were ever. The biology is still very important in the process, despite taking a second seat next to the automation and the computer sciences that are involved in the process. And despite being more complex and more multi-processed, we are now coming with drugs more rapidly than we ever did, and these drugs are better and better and safer and safer.
Thank you, Tirosh. We do have a number of questions now in our queue. This is a fascinating presentation. I wish I had paid more attention in my biology classes in school. One of the first questions that comes up here is, well, I'm going to ask a question first because you mentioned it. So you talk about FDA approval. We know that there are other countries that are working on drugs as well, China being one of them. Does everything run through the FDA in the United States, or is it possible there could be drugs that are developed outside of that review process?
Right, that's an excellent question. And yes, there are. And nobody is going to stop the Chinese government for deciding that this is a compound that we want, and this is a compound that we're going to give to our population. However, just like the FDA was just about to approve hydroxychloroquine and then pulled that recommendation, even the Chinese government and even the Russian government and any government who is not playing nice with the FDA are very aware of the quality of the review of the FDA, and in all the cases that have happened so far, tend to align themselves with the FDA, especially since it's very, very difficult to get all the participants that are needed for the phase 1, phase 2, phase 3 clinical trials. Luckily for us, it doesn't feel that way, but there's not too many SARS-CoV-2 patients to participate in these trials, and there is a lot of potential treatments for SARS-CoV-2, which makes it very hard to gather scientific information. Now, can non-scientific information, and I'm not saying that what is not FDA approved is not scientific, but the FDA has definitely set a very good standard to the whole world of what is good review processing of drug discovery. Yes, it can happen, but from experience, from looking at history, it rarely happens.
Got it. So we do have a number of questions in our queue. I'm going to try to go through them as quickly as we can, because I know you have more slides to present as well. So one of the first questions is, I lost it here for a second. So this comes from Carl Rothschild, who's a physician, and he asks, I'm aware that there are many viral diseases that appear to decrease in virulence over time. Does this seem to look like a similar situation with COVID-2, with SARS-CoV-2?
Yes, we have some fascinating genetic information that shows that unlike what we expected, SARS-CoV-2 doesn't change as much as other viruses of the same type. That means that it's likely going to be as infective as it was in the beginning if you would expose two people who never saw the virus. However, at the same time, you do have antibodies and an immune response that protect you from infectious agents every day. And stronger immune systems are always correlated with stronger protection from infectious diseases, including SARS-CoV-2. So any infection over time would seem less infective if we do not account for the resistance in the population. From our genetic evidence of SARS-CoV-2 changes, we don't see any reason to believe at the moment that SARS-CoV-2 is becoming more infective or less infective. However, genetics and biology usually stress is what is driving evolution. And I kind of alluded at that when I was talking about drugs. So if you stress the virus population by treating them with drugs, then that is when evolution will strike fastest. And that is when you can expect to see the most changes in SARS-CoV-2. Unfortunately, we haven't come up with a treatment yet, so we haven't been seeing that in the genetic data.
Got it. This next question seems a rather technical one, but it may be relevant to what you're discussing. So the questioner asks, I understand using culture methods for looking at antibacterial compounds. What are you using to screen for antiviral activity? Tissue culture or something else?
Yeah, I love that question because I've prepared a complete set of slides about it, which I'll get to when I discuss my research. But in short, I'll spoil the surprise, but we use mini organs. We grow tissues in a petri dish, we grow brains, we grow livers, we grow intestines, derived from embryonic stem cells, and we use them as models for viral infection.
Got it, thank you. There's a few more questions here, both about the economics of treatment and the virus. I'm going to save those for later and let you continue with your research slides at this time. Okay.
Okay. So thanks for the questions. Now we'll discuss a bit about what is happening at UBC, which previously a lot of the information that I talked about was just to bring us up to speed. I'm sure a lot of you have heard most of what I was discussing already if you've been tuned into the news. But UBC and its facilities are quite unique, and part of the reasons why we are one of the first labs in Canada and in the world who can do drug discovery against SARS-CoV-2. So UBC has long had the facility for infectious disease and epidemiological research, which is one of the facilities that can deal with contamination level-free organisms. That is tuberculosis, that is some strains of influenza, West Nile virus, but that is also SARS-CoV-2. And Finder, as we call it here, is the largest and most advanced contamination level-free facility associated with universities. Obviously, drug companies have their own fancy rooms, but in a university settings, Finder is one of the most advanced and expansive facilities that are in Northern America and in the world. So we have those facilities here with a lot of equipment that is needed for this research. On top of that, UBC and the Life Science Institute have automated workstations in our high throughput automated facilities, which can handle the large workload of sorting through tens of thousands of compounds in a way that doesn't introduce human error. So even if I'm the most vigilant researcher in the world, after handling 100 compounds or 400 compounds or 4,000 compounds, there's a big chance that I'll introduce some error. And these automated workstations allow us to do it without that. And also when we're looking at combination therapy, which is acutely important for developing treatment against SARS-CoV-2, generating all the different mixes of all the different drugs in different concentrations is a task that if it was given to a researcher checking it on the bench would take many lifetimes. But with this workstation, we can do it in a matter of days. Also, UBC has Finder skilled researchers, which is one of them is pictured here during work. That is me wearing an N95 mask before it was cool. We also have screening microscopes, which images I showed you earlier, images derived from it, that can very quickly, in less than 30 minutes per plate, and each plate has between 100 to 500 different compounds and compound combinations on it, can go through plate by plate by plate. And of course, we can connect it to our automated workstation and leave it there for the weekend and come back and have a result of our screen and our drug combinations. UBC also has many researchers, skilled researchers looking at the social, economical, and biological impacts of SARS-CoV-2. There's a full link here if you want to get more information about each one of these researchers and what they're doing and how they're researching the impacts of SARS-CoV-2. Out of these 450, sadly, there's only 2 virologists. One of them is Dr. Francois Jean, my boss, who has also founded Finder. This is his baby. And we recently received a lot of support from the Canadian government with grant money in order to conduct this research, this drug discovery. And we're also collaborating and enjoying collaborating with Dr. Eric Jean, who's another virologist working at UBC, which is also funded by the Canadian government for this research. And together we have a very formidable team looking at the biology and developing drugs against this pathogen. So here is, I was hoping it would be a magic trick, but I already gave it. So I probably shouldn't leave science to be a magician. But the uniqueness and the reason why we were granted so much funds is because we have a very unique testing system. We test lab-grown organs inside a dish, and here is a picture of some, in this case, mini brains that we grow in a dish that we are generating with our collaborators in stem cell technology, another Vancouver-based company that is doing very important research on SARS-CoV-2 with us. These mini brains allow us a lot of power when we're screening these drugs, because usually when you test a certain infection, you're only testing it against one type of cell or 2 types of cells. But we know that the human body is more complex than 1 cell or 2 cells. And the virus might infect one type of cell in your body, and a drug might be effective against the virus in that one type of cell, but it can completely escape treating the virus in other cells and in more complex organs. This is also very important because of the ability of the compound to get into the cells where it is needed. So when we're screening our compounds against these as close as possible to humans, in a dish, then we can also see which compounds can easily get in there and do the job that is needed of them. And we've already used this model successfully when we were tackling the last outbreak of the Zika virus, which also infects brains. Of course, SARS-CoV-2 infects many organs in the body, not just brains. We're also investigating lungs in culture, kidneys in culture, and we're looking at which drug combination is more effective against each type. And obviously, we will want a drug combination that is effective against all of them. Using organoids, as we do these organs in a dish, we can also overcome this complication of not having mice models that are capable of replicating the disease as it appears in humans. And part of having this slightly more expensive, slightly more difficult to handle system, it also means that we will be saving a lot of time once we discover treatment to go straight into clinical trials. And what we're doing, as I showed with the example from tuberculosis, we're just putting thousands of wells with thousands of compounds, and then each one in combination with each other. And these compounds we're getting from collaborators from all over the world. We have labs in Brazil who are sending us natural products, labs from Tunis, labs from Russia. We have collaborators that are developing their own compounds based on previous antivirals. And because we have this unique system, everybody wants us to test their compounds, which we're very happy to do, because when you're screening 10,000 compounds, adding another compound is a very, very simple process. Yeah, so that's the end of my spiel. So I wanted to thank you for your time and open it up for questions by the audience. I also had a couple of questions emailed to me that I'll be happy to discuss in case People are shy, but I don't think that's the case.
Thank you, Tirosh. Thank you. And I know we're short on time. I want to dive into maybe the big question, which is, are we going to find a treatment or a vaccine, and when?
Okay, so the answer is yes, we are. We are going to find a treatment or a vaccine. We've already found a treatment. Remdesivir is acting very well against severe cases of SARS-CoV-2. However, it is not enough because we want to treat everyone. When? That is a good question. Looking at SARS-CoV-1, the only reason we don't have a treatment now is because SARS-CoV-1 disappeared and it was no longer sexy to keep on studying it. And if we had, we would be better positioned now to treat it. But in the lab, we can find it in 6 months, easy. Then through clinical trials, earliest that it will get into patients that need it will be 3 months to recruit the cohort. So we're looking at 9 months before any compound is going to get into patients who need it.
So just to build on your answer there, thank you. So SARS-CoV-1 disappeared because it ran out of hosts, essentially. I mean, it wasn't allowed to break out as fully as COVID-2 has.
Right.
So is my understanding then that this is not going to disappear on its own, unless the entire population were being affected?
SARS-CoV-2 would not disappear on its own, but we are seeing how strong the epidemiological control of it is. We are seeing a great ability of very extreme but effective epidemiological techniques like curfews, like closing down businesses partially or fully, are having a huge effect on the disappearance of new cases. So what will happen is that we will have the virus going with us for the next couple of years. Most of the people will not have it, but the people who will have it will be isolated, identified well, and then hopefully when we have a drug, we'll be able to also make sure that they get cured.
So a couple of questions that have come in. So a lot of viruses did not have effective medicine until vaccines were found, polio, chickenpox, etc. And this questioner wants to know, why is there so much focus on finding drugs to treat instead of finding a vaccine?
Right. So the main answer is because viruses of the same family were never successfully vaccinated against. So we don't have good vaccines against the common cold, we don't have good vaccines against SARS-CoV-1, and we don't have them against Middle East Respiratory Syndrome virus. It doesn't mean we can't get them. It just means that we're more likely to get drugs rather than vaccines. Part of it is also the ability of the virus to mutate that we discussed, which is stronger when the virus is trying to deal with a vaccine compared to when dealing with drugs or drug cocktails, definitely.
So can you expand a little bit on what you think the world looks like until we get to that treatment or vaccine level that you're talking about? I mean, are we social distancing for the next 2 years? And
Okay,
masks and all those things?
Now you're asking a biologist something that you should probably ask Dr. Boni, but the answer is yes, we'll see very gradual openings and we'll see restrictions being kept. We'll see some countries like the United States that have large instances of the virus being kept closed. They can do what they want inside the country, but other countries will not allow travel and access from countries that have large instances, because nobody wants to erase all the difficult but meaningful progress that we've done so far. So the outcome of these processes is that less and less people will be infected. It'll be easier to identify and isolate clusters that do outbreak and isolate them from the rest of society. So the way the world looks like, in my opinion, in the next 6 months is gradual reopening. And then for the next year, very close attention, keeping on monitoring, maybe special apps that track you and know who you have been in contact with. And then in case there is interaction and there is a case of disease emerging, a very quick and rapid activity by, by health officials to test all of those who you've been in contact with. So I think for the next year and a half, maybe, knowing who your circles are and having good trace of them is going to be a very important part of moving out of this this pandemic.
Well, that's a very sobering analysis. I don't think it's any different than what most of us are hearing, but I think many of us are sitting there hoping that as Dr. Henry says, it's not going to be a light switch, but I think many of us are hoping that somebody's going to flip a switch one day and then we're going to go back to something. It doesn't sound like that's in the office.
And that will happen when we get a drug, when we get a treatment or a vaccine. You just pop a pill like you do if you go to travel to northern Thailand and there's malaria there. You take anti-malarial drugs and most of the times you'll be fine. And that is where we will aim to be in about 2.5 years from now, when drugs are verified by FDA approved and manufactured to an amount that is able to be delivered to everybody in need.
So do we think that what's going to happen is you're going to get sick, drug will get you better, and then like the common cold or the flu, you're going to get sick again and you'll take the drug and get better?
Exactly that. Until humans develop innate resistance to the virus, that's exactly what we're going to see.
And how long do you think that innate resistance would take?
The current information that we have is that only about 10% of the population has any kinds of signs that they've experienced the virus, and those who have might be reinfected. So being immune or being exposed doesn't make you automatically immune, which complicates things. That's a less sobering message, is that we can develop resistance, and we have developed resistance against a lot of viruses, some of them we never heard about because that happened 10,000 years ago. So that can happen. The rate of that, we're looking at evolutionary rates, we're looking at, of the virus, we're looking at years, 2 years, 3 years, 4 years, at least.
Mm-hmm. We're over our time. You've been phenomenal. You've pretty much answered all the questions that remained, and as you indicated, some of them are really probably questions for our politicians or our, maybe Dr. Henry, about the economics of this and the politics of preparing for the next epidemic. Is there anything that, I guess I'll ask one last question, then I'll ask you to leave us with your final thoughts, which is, one, we have a very low instance, it seems, of the virus here in BC. Dr. Henry, Dr. Dix, and all of our healthcare professionals and frontline workers, Dr. Patty Daly, who's a member of our congregation, have really done a remarkable and heroic job. And so have the people of Vancouver and British Columbia in general in following their instructions. How safe do you feel going out and about in Vancouver today?
Not very safe. I drive from East Vancouver through Kitsilano Beach to get to UBC, and seeing how people go to bars and the distance in which people are sitting from each other on the beach, it doesn't make me safe. It doesn't make me feel safe, and it will cause reemergences of the virus. Is that a disaster? No, it's not. So we will allow, infections will happen as much as we want to control them, they will happen. I personally haven't been going to bars, and if I would probably take a mask with me if I know that I cannot keep safe distance from other people. Same goes for grocery stores. It's very likely that you won't be able to keep distance in a grocery store. So protecting yourselves by following the advice of these great professionals, as you described, that's the key. As I said, it's all about epidemiology control. But the human nature is quite predictable. If you drive on the highway and there is something slowing traffic, people tend to speed up right after they leave that area. People have fatigue. People are happy about the progress that we made, and they should be. But people also feel very confident. And that will lead to more instances of infection. Again, it's not too worrisome. We have an excellent medical community here that has been remarkably treating patients who do get infected. And as you said, BC has excelled in treating COVID patients and mitigating the risks. So I can't afford getting sick right now. I have stuff to do.
We can't afford you.
Thanks. I mitigate the risks. I have friends that I would love to meet and grab a drink with, but we'll just have to wait.
Tirosh, we are so blessed and fortunate to have you and your colleagues working on this. To know this happened in our own backyard is in its own way such a blessing that UBC is outfitted for this kind of thing, and that we're using those incredible facilities. Any final words for us?
What I want to say is I'm part of this community, and I'll be happy to be available for future questions. Like I know some people might have not had their questions answered. People might have other questions, or having heard me discuss it with family members and friends, they might have questions. You guys have my email, and feel very free to use it. I won't answer always immediately, but I will answer to the extent of my capabilities. And again, as a biological researcher, definitely not as a medical professional, but I'll be very happy to keep on taking questions about this from the Temple Sholom congregate.
Tirosh, thank you so much. I'm so glad that we could set this up. I want to thank all those that joined us online. This entire program has been made possible by our caring community fund at Temple Sholom. Our members have donated to help offset the cost of things like the Zoom technology, and also to make sure the temple can weather this economic storm as well as this health storm. We'll share this online on YouTube and on our website for those that didn't get a chance to see it, or for those that have seen it and want to share it with others. You've been just wonderful. I wish I could invite you over for Shabbat dinner. It would be, you wouldn't have time to eat because we'd be asking you so many questions. But I hope one day
I haven't shaved. I'm blushing here, so.
That's okay. At least you have hair to cut. I don't even have that. Thank you so very much. You're just fantastic.
It's been my pleasure.
The most informative discussion. Thank you, everyone. Thank you for joining us. Services tomorrow night are at 8:15 online, on TELUS, on YouTube, on our temple website, on Facebook. There's a family service also in the park for those that have young children ahead of that at 6 o'clock. Thank you again, Tirosh, and to everyone. Have a wonderful weekend. Enjoy the sunshine while it lasts, and an early Shabbat Shalom.
Thank you.
Thank you.
Thank you, Rabbi Dan, and thank you, everybody, for organizing this.
Thank you.